Everybody to this session on medical text and data mining. My name is David Suter and I've been given the honour of chairing this session, and our first speaker is Robert Hoehndorf from King Abdullah University of Science and Technology in Saudi Arabia. Without further ado, I'll turn it over to Robert.
So thank you very much. It's my great pleasure to be invited to speak here, and I'm going to talk a bit about our work over the past 7 years that I have been at KAUST in Saudi Arabia, working on machine learning, biomedical ontologies, and applications in precision health. So we have seen a similar figure yesterday in many talks of this workshop. We know that biology and biomedicine are very data-driven disciplines, and we can get very large amounts of data on different levels of granularity. And we can not only get this for cohorts or in some research projects, but increasingly we can get this information also in a clinical setting for individual samples or individual patients, ranging from genomics data over transcriptome, proteome to the metabolome and then the phenotype. And one of the challenge, and this is something that we don't only get for particular tissues or single samples, but now we can also get this for single cells and for multiple cells within an individual through single cell sequencing and single cell technologies. And one challenge is how to combine these different types of data so that they make sense. And these are these little connections and arrows in this figure. And most of my talk will be about these little arrows which connect the different data types together that we can get from single samples or from eventually from individual cells, so that we can combine this and then use this for machine learning and to answer some specific questions. So while biology is a data-driven discipline, we need to ask, where do these little arrows here that provide the connection between the different levels of granularity come from, and how are they represented? We will see that biology or biomedicine, not only data-driven disciplines, but also knowledge-driven disciplines, because these connections between the different data types and between the entities which are being measured and which we can measure through high throughput experiments or targeted experiments, these connections, they come often from experiments. So it might be that to connect these, we rely on experiments of 1,000 different PhD students which have studied one interaction or one protein or one gene or one transcriptional regulation for their PhD in a lab, then published a few papers and their thesis. And in the end, we know now that, for example, FOXP2 binds to COX1, or that a protein has a particular function. And these are small micro theories which we represent in biology and biomedicine in knowledge bases or knowledge graphs, if we use a graph-based representation format. If we have some quantitative information, we would represent them in systems biology models where we model the dynamics or the quantitative relations between these entities. But essentially, these are judgments which result from experiments. They are supported by evidence, and they are not easily accessible or not directly accessible from the data that we see. So we have to, when we measure, we determine a genome or a transcriptome from one sample, then we have to situate our data that we see with respect to this background knowledge that relies on experiments, sometimes of decades of biology. A second type of knowledge that we rely on is a priori necessarily true knowledge that defines our terminology or the concepts and how they relate to each other in that we use to express our findings. So you can see that FOXP2 has a function of macrophage apoptosis. I'm not sure if it's true, it's an example here, but macrophage apoptosis is a biological function or a process that a priori we can define using axioms. These axioms occur like this in the gene ontology. So macrophage apoptosis is equivalent to an apoptotic process which occurs in a macrophage. Okay, this is maybe obvious, but then we can go on from there and look at another axiom which restricts macrophage, and a macrophage is capable of some phagocytosis, which is another biological process. And then we have, just based on the terminology of what it means to be a macrophage apoptosis, not only established a relation to macrophages, for example, a type of cell, which is a type of cell, but also what these macrophages or the processes that these macrophages usually are involved in, so things like phagocytosis. And when we explore, when we build models in biomedicine, we need to find a way to combine this. So we have big data on different levels of granularity, ranging from genotype to phenotype, also including the environment. We need to situate this data that we measure against very large amounts of knowledge, prior knowledge, which allows us to interpret and make sense of this data and to connect the different data types that we identify together in a meaningful way that is consistent with experimental results. And then we need to find a way to use this combination of big data and big knowledge for some machine learning or optimization, or essentially some intelligent decision-making process. And this is what my talk is going to be about today. So this is a schema of what I'm going to talk about. So we'll start with symbolic representations of our knowledge. This might be knowledge graphs, it might be knowledge bases, and biomedical ontologies. So I call these symbol systems because they include not only, they allow us to reason over this knowledge and draw inferences. And on the other side, we have data, things that we measure. Often these are quantitative. This is quantitative information. So it might be genotype when we do sequencing. It might be gene expression for transcriptomics experiment, and many more. It might be images. And we want to find a way to connect it. And this is a representation of the neurosymbolic cycle, where we take our symbolic knowledge and embed it in a space where we can combine it with the data, where machine learning and optimization takes place. And then from this space, we want to extract, again, symbolic representations so that we can determine whether the decisions that our agents make, the results of our learning process, are consistent with the knowledge that we have about a domain, so that we can identify or eliminate inconsistent knowledge, or we can revise our theories. And I do this in two parts. So the first part is I want to talk about embedding methods, specifically for biomedical knowledge bases, including ontologies. And embeddings, and I like to define this first. So an embedding is a morphism from one mathematical structure into another structure, Y. So it's a structure preserving map between two mathematical structures. And the aim of having an embedding is to preserve the properties of the first structure in a second, but the second structure may be more useful for some operations. In particular, we can, for example, we may have some natural ways to determine similarity in the second structure, or we can use gradient-based optimization in the second structure. And the key question here is which aspects of the first structure, so what is this first structure X, and which aspects do we want to preserve in, which aspects of Y do we want, of X do we want to preserve in Y? And in general, we want to embed knowledge bases in some Rn, mainly because this is useful for gradient-based optimization in many of our machine learning models. And I will just briefly mention three approaches: graph-based, syntactic, and model theoretic approaches for embedding ontologies. And the first one, graph-based approaches, is actually not entirely, so many of you will be familiar with knowledge graph embeddings. And the key question is, how can we take an ontology and description logic or OWL ontology and convert it into a graph? So there are some parts of ontologies which are very easy to convert into a graph, and it's very common. So subclass relations, we simply create an edge x, a directed edge between x and y, x is a y. We can take some existential restrictions which also give rise naturally to a graph-based representation, such as here this constraint that x is a subclass of part of some y. Same for regulates, and then there are some which are a bit more challenging. So x and y are disjoint. So if x and y are disjoint, need some thought of how best to represent this in a graph-based form. Same for equivalence. There are different alternatives. So x is equivalent to y, so two classes are equivalent, and maybe there's an edge between them with a particular label, or we just create a supernode where we merge x and y. And the second challenge here is that how to do, so these graphs can be based either just on axioms, so basically the representation as the ontology is written down by its designers, or it can be based on inference, so include some deductive mechanisms for inferring knowledge. So here we have, if x is a part of some y and y is a subclass of part of some z, and part of is transitive, then we would infer that x is also a part of some z. And this is something that is not asserted in the ontology. It would require access to some reasoning or inference mechanism. And we tried to test this with some simple graphs. So I basically just then, when we create this graph, so here, this is one way to represent some A box plus T box statements. So a knowledge graph, FoxP2 binds to map K3, for example, together with some ontologies. So these are the two classes, these two Go classes, about which are functions of proteins. There on the top left. And the dotted line, so the dotted dashed line, red line, this is indicating an inferred relation. So this is something that would be inferred deductively, and we may or may not add this to our graph. And then we can use a graph embedding, for example, one based on random walks, in which we have sentences, and then embed this through a language model, or one of the many other knowledge graph embedding methods that have been developed in the past. And these embeddings we can then use for different types of tasks, such as, for example, edge prediction. And we tried this in 2017 on a large knowledge base consisting of several ontologies and an ontology of phenotypes, diseases, and functions. And we tried to predict a number of different relations between entities which are relevant in biomedicine. So we tried this on 8 different relation types, and it was very surprising to us at this time. It was, at the time, one of the first applications of knowledge graph embeddings for biomedical knowledge graphs and discovery of new relations. And we found that we could basically match the performance of many state-of-the-art methods basically using only this. But there's a second thing, and this is more the topic today. So there is this distinction when you look at these performances here for the different edge types. We compared here what is the performance without this deductive closure, so without using any reasoning, and using reasoning over this ontology together with the knowledge base. And my hope was really, if we apply a reasoner, we should have more access to more information, and therefore the performance should also increase. And when you see here, that is definitely not the case. So the performance is basically the same, sometimes even it drops a bit when we apply this deductive closure to this graph. So this was a bit disappointing, despite all the other nice things that this method gave us. It was a bit disappointing that we could not actually gain an improvement in performance using the axioms of the ontology and using inference. So then the question was, is there maybe something wrong with our method, and is there something that we could do a bit better here? And one thing that we tried to do is then, well, we tried to go back and said, okay, what if we just don't use any knowledge graph embeddings, which are graph-based, but we simply embed axioms directly. This is nice because there are some axioms in gene ontology which we simply cannot represent as a graph very easily. For example, the distinctions between universal and existential quantifiers are very difficult to capture in a graph-based form. And some more complex axioms, so where, for example, cell proliferation and things which occur in fungi are disjoint. So these things are quite difficult to capture directly in a graph. So the question was, can we embed these axioms directly? And before we did that, we looked a bit more deeply into ontologies, and ontologies, they contain more than just axioms. They also have labels, synonyms, definitions, authors, a lot of metadata information. So things like macrophage, apoptosis, for example, these are labels not primarily intended to capture semantics, but rather to be useful for people who use this ontology. And we also use some method from transfer learning where we tried to train a large language model on PubMed or on biomedical art, and then apply this to ontologies. In the end, we built a method called ontologies plus annotations to vec. And this is a rough overview of how this method works. So we have two corpora. One is based on the axioms in the ontologies, and it contains T-box and A-box axioms. So T-box axioms are the axioms that constrain classes. The A-box axioms, in my mind, are mainly what we would represent in a graph-based format, but these are essentially facts or relations, binary relation assertions. And we will generate a corpus from the axioms, and we generate a separate corpus from all the natural language data in these ontologies. So this might include the label, it may created this class, so who edited the definitions and the axioms, what are the synonyms, and so on. So then we apply an automated reasoner, either Hermit or ELK, depending on the type of ontology, and we do apply a deductive closure here, use our pre-trained model on PubMed, and train a word-to-vec or a language model here. And in the end, we get an embedding for the classes and instances in our knowledge base, which combines information in the axioms and the metadata. And we tested this on a relatively popular benchmark to predict protein-protein interactions based on functional similarity in the gene ontology. And our baseline method, and this was the state of the art at this point, was this Resnick semantic similarity measure. And this was performing quite well, and you get a ROC AOC of about 0.8, roughly in human and yeast for this. And this was something that was very interesting, using only the axioms and just a cosine similarity. We did not improve upon this. But then using the labels, synonyms, and definitions of the classes, even just using a cosine similarity, we got a huge improvement from about from 0.79 to about 0.87 in human interactions, similar improvement in the yeast. And then we could also train this using some instances. So this is a kind of similarity learning where this method becomes a kind of trainable semantic similarity measure over the ontologies. And this would improve it even further. And we could improve on this benchmark by almost 0.1 AOC, over 0.1 AOC. This was nice. And at this point, we found that semantic similarity measure, or this would work as a trainable semantic similarity measure over ontologies. And this is something that is applicable to a very large class of problems in bioinformatics. There was still something that we were not happy with at the time. And this is that this is essentially a relatively a method that ignores all the semantics of the axioms in our ontologies. It's only based on word co-occurrences because it uses word to back. And the question is, are we able to come up with an embedding method that actually captures this semantics? And when I talk about semantics, I mean the model theoretic semantics of the ontologies that we use in biomedical applications. So this is the semantics, the formal semantics of a relatively lightweight description logic called EL or EL+. And you can see that there is a syntax and then there is a semantics, which is an interpretation within a sigma algebra, similar to first order logic. And the question that we had is whether there's a way to preserve this relation between syntax and semantics as part of these models. And our idea was, if we have a knowledge base T with a signature sigma, so our knowledge base is our ontology together with the A-box statements, then we want to find a function, an embedding function, which maps the symbols in our signature into the Rn such that the image of this function is a model of our theory T. So this, if we find this, then we would have an embedding which exactly preserves the model theoretic semantics of our theory. And we know that at least in theory, such a function should exist because we know that EL and EL++ theories do have infinite models. So all of them, if they are consistent, they will have infinite models. And we know that any consistent EL++ theory will then also have a model in the Rn based on the theorem of Löwenheim's golem in the upwards direction. And our idea was then we define two functions. Essentially, we map concepts to n-balls, so open n-balls, so that our embedding of a concept is the set of points which lie within this open n-ball. And this is going to be the extension of unary predicates or concepts within an Rn. And for relations, so binary predicates, we simply use the trans-E property, where we have relations as a transformation between 2 points. In order to find this function, this embedding function, we use the axioms in our theory as constraints. And one advantage that EL has is that we only have to focus on a particular small set of normal forms, so 4 normal forms, to which any knowledge base can be converted. And the way in which this works is, so I just give you an example here. So the way this works is we have our theory on one side, and here we have our embedding, and you can see these extensions. So this red circle here, this small red circle is a male, and you can see that this this optimization will end when it lies entirely within the person circle. This is this yellow circle here, based on the first axiom. Similarly, for female only, it becomes much smaller because it already is quite small in the beginning in this case. And in the end, we also can show that if this optimization procedure terminates with a loss of 0, then the resulting embedding is indeed a model of our theory. Okay, so this was about the embeddings of ontology axioms, and essentially this is a set of neural embedding methods specifically for description logic axioms. They can be based on graphs or converting axioms in ontologies to a graph. They can be based on the syntax itself, or they can utilize the model theoretic semantics. They are applicable to semantic web ontologies, description logic ontologies, which is the main language that we use in biomedicine to represent ontologies, and they include knowledge graphs, because knowledge graphs are essentially a boxes in these description logic theories. And we have some proofs of soundness and some basic meta theory of these embeddings as well, which translates from the meta theory that we have from description logics. But this is now only a way to embed the ontology axioms, but I have not talked about data or combining this with any biomedical data so far. So the second part, and before I go to how to put it all together, I'll show you the second part of this neurosymbolic cycle, and this is the extraction part. What if we have some data, any kind of data, and we want to have something that is consistent with our symbolic knowledge, so consistent with our ontologies and other information that we have formally represented. For this, we need to have a way to extract symbols and then be able to reason over these symbolic representations. And I'm going to talk about the case of protein function prediction first, and then we can put it all together so that we can see this whole cycle. So for in general, what we want to do in the extraction part is to generate symbolic representations which are interpretable because they're based on symbols, they should be transferable, and we need to be able to apply them in automated reasoning so that we can test consistency of what we have found. And we can do this for a number of applications. I'll start with protein functions. So the aim of protein function prediction is given a protein sequence, which we can determine either through, either we can predict it or we can sequence it directly and determine the sequence of a protein. So given a protein sequence, we want to know what the function of this protein sequence is. And the function of a protein sequence is represented using the gene ontology, which is ontology of about 45,000 classes and over 100,000 description logic axioms. It's one of the largest ontologies that we have. So prediction must be of a protein function must be consistent with the gene ontology. And we have built a number of different models, DeepGO and different variations of this DeepGO model. So it's also now 4 years old, DeepGO, specifically to output predictions of protein functions which are consistent with the gene ontology. And they take mainly as an input the protein sequence. And you can see there's an embedding part, so there's a network embedding here. I will ignore this for now, but you can see that there is the second part. I will not, so the first part, the embedding part, I'm not going to focus on at this point. But I want to talk about the second part, the prediction of symbolic knowledge, so that we can determine consistency. So what we have built in DeepGO is a neurosymbolic classifier which uses the axioms of the gene ontology as constraints. And this classifier is based on a kind of fuzzy logic called Lukasiewicz-Tarski logic, where we translate some of the axioms in gene ontology into constraints on this optimization problem when we train this DeepGO model. So essentially, we will have a small layer for each class in Go. So this is a small layer consisting of fully connected layer plus a particular function which takes the maximum of all its subclasses. But essentially, it is a way in which we implement a constraint coming from this Lukasiewicz-Tarski logic, derived from subclass relations. We can also do this for some existential quantifications in gene ontology. I didn't show this here. in particular regulates relations and partout relations. We get one thing for free, which is a hierarchical refinement of the features based on the structure of the gene ontology, specifically the taxonomy of the gene ontology. When we look at this, we'll find that while DeepGO is not really the very best model in some competitions, so here on the left-hand side you can see we participated in some biannual competition now, the second time we competed in this, where we compared DeepGO with a number of other function prediction methods. And it is always amongst the top ranked methods. There are a number of different sub-analyses we can do. Maybe not the very best in terms of performance, but it has a number of great advantages. It relies only on the protein sequence as an input. So it is something that where it is universally applicable. We don't need protein structures. We don't need to predict protein structures. It is very fast, so it can do something like 50 protein sequences per second on a single GPU. So people use this in a high throughput manner when they want to functionally characterize metagenomes or newly sequenced organisms. And we also have a web service for quite some time running, where over 500,000 jobs have been completed. Some jobs are very large, some are very small. But there is over 1 million protein functions have been predicted using DeepGO for over 1 million proteins. And this is very useful, and the key advantage here, it was also one of the first deep learning models for protein function predictions, DeepGO, but the key advantage is the enforcement of these consistency constraints with respect to the gene ontology. Okay, now this is one way in which we can output hierarchical or symbolic representations using this DeepGO classifier. Now let's try to put this a bit together. And I want to get back to actual biomedicine, where we do put most of these methods together. And I have two cases. The first one is rare disease, rare Mendelian disease. The second one will be cancer. And in Mendelian disease, or rare genetically based diseases, we have used our methods to build a number of prediction methods for which identify causative or causal variants in individual genomes or whole exomes. And this is, so which variant in a human genome causes a set of phenotypes is very difficult to identify, because we need to know how this variant will affect the function of the protein, if it is protein coding, otherwise how does it affect regulation of a gene, or what is the functional effect of this variant. We need to identify what are the functions of this gene. From there we can aim to predict what pathways, networks, and organismal functions would be affected. Then how is the physiology of this individual affected, and are there any particular interactions with the environment which would be affected by this variant. And none of these questions can be answered from data alone. It relies on background knowledge, and in this case a large amount of background knowledge, in order to determine the functional and in the end the phenotypic effect of a single genomic variant. And we have been building a number of different tools for this. So mainly under this, we call this, it's our PhenomNet project, so where we have the PhenomNet variant predictor and a number of different variations on this for different types of variants using our PhenomNet ontology. And the idea is that we take background knowledge, formalized background knowledge graphs plus ontologies from a large number of model organism databases and databases from functional genomics. We combine it with personalized information, in particular genome sequencing data, so the genome or the set of variants identified in one individual might be based on a panel, based on an exome, based on a whole genome. And we combine this in one of our variant prediction methods in order to predict the candidate disease-causing variant in this individual. I forgot there's one additional input, and this is the actual phenotype observed in this individual encoded using ontologies. I just show some of these rankings based on some comparison against a number of different methods. So we can identify this in over 70% of the cases. We can identify these disease-causing variants, which is quite a lot better than a number of other methods that we compared against and other people have compared against. And our key advantage here is that we are better at using background knowledge. So we use in general more background knowledge than most of the other methods. And the second advantage is that we have better ways of exploiting this background knowledge in our machine learning methods. And I show you one example here. So this is an example. It's a specific model, but it's very similar in many of our methods. So this is a system that my student Asa has built for copy number variants. And we take as an input a set of phenotypes and the set of variants. So the phenotypes tell us what is actually observed clinically in one individual, and the set of variants are the set of genomic variants in this individual. So this is the input, and this is something that is obtainable, especially for rare disease patients in a clinical setting. The phenotypes we use in our knowledge base, where we combine ontologies together with a set of annotations and the interactions between different genes or their products. So we build a knowledge base which we embed here using a method that we developed specifically for this called DL2VEC. I didn't specifically discuss this method before, but it's an embedding method for ontologies. We found it to work for this kind of problem better, so we get a representation of the individual patient, so based on only the phenotypes of this patient, and we get representation of our background knowledge, so we get embeddings for genes, and we get embeddings for our patient using this DL2VEC method. And now we have taken our symbolic representations, projected them into a space where we can combine it with our data. So this is then the bottom part B of this model. So this model, there's a pre-processing happening on the genomic variants that we see. So there's some annotation from public databases where some features are added to these variants. These might be, where is this variant? What kind of genes does this variant overlap? Is it in a coding region? Is it in an intergenic region? And so on. We embed those as well into a space. So these features are embedded in a space where they can simply be concatenated with these phenotypic representations that we have generated using the first model. And then in this space, we then have a representation both of a genomic variant together with the background knowledge about the variant and about the patient. And we can use this in a model in order to predict copy number variants which may cause a disease. And you can imagine that this is very similar for other types of variants. Although this is a model for copy number variants, we can take other types of variants such as indels or single nucleotide variants and replace how we annotate these variants at the bottom part and still combine them with the background knowledge in the same space, then have a model that has access both to the background knowledge about the variant and the actual information about which variants are present in order to predict and rank candidate disease-causing variants. So this is the general method, or it's a general method for identifying rare disease variants. And we have a number of different systems for this. I think over time, we add different features and different components to this or generalize this model further. There's one question that I still want to discuss, and that is interpretability of our models. that is, of course, very important. And many times it was mentioned, or several times it was mentioned yesterday. And the question is, what does interpretability of our model mean here? So maybe it's an explanation of the decision based on our background knowledge, so the graph or the knowledge base combined with the data. If we want to have such an explanation, it may include ontologies. And we have a different application, and we're a different application area that I'm going to show on cancer, where we try to gain some interpretability, and I show you what we can get from this using graph attention. And this is a similar case. So here we have not only a PPI network, but we have a PPI, a protein-protein interaction network together with background information. So this is in general. We also have ontologies in here. And then we project different types of omics data onto nodes in this graph. And the idea is that if we measure gene expression, then the transcript would be a node in this graph, and we assign it a feature based on what we have measured for gene expression. Or for methylation. Or if there are genomic variants, maybe we assign all of them as a feature, or we just assign some kind of extracted feature, such as the allele frequency of this variant, or maybe the rarest variant, or a burden test for each gene. Based on these variants, we can assign these as features to nodes in this graph. We can then use a graph convolutional neural network if we have this graph-based representation together with an attention mechanism where we can use self-attention pooling. Again, we can use graph convolution and so on. And here, what we are building is a model that predicts survival. So it's a prognostic model, or it's a survival model for cancer. So here, our data are multi-omics features derived from individual samples. So we annotate this graph. One instance is the kind of omics features observed in a single sample. These are the features for the nodes in the graph. The output is survival risk or survival time for this individual. We can similarly embed some kind of metadata. So this is part C. So we embed this metadata in the same space. This is some other metadata coming also from ontologies. We can embed this and then use this in a dense layer. We can combine this in the end as part of the same model. Now, this is all fine. We have done this we have built this as a pan-cancer model, where we use transfer learning between the different cancer types, and in some sense, we also do so it generalizes between different cancer types. The cancer type is an input of this model, and we use graph attention to focus on and then rank particular nodes. This is part of this model, and in this part we rank them and then do a pooling based on the top k ranking nodes in order to reduce the size of the graph. And this attention rank depends on the input features. So it's personalized because the input features for one sample are derived from one individual patient, so from one tumor. And the question is, what does this model actually focus on if we train it this way? So what nodes would be active in such a model? Are these prognostic markers, drivers, or if we look at the ontologies, would these be more functions or phenotypes? And when we look at this, we have done this, and this is a pan-cancer model, so it works for all. I just give you some glimpse. You can look at the full paper for all the results. But in breast cancer, in 89% of our samples, the top ranking node is a known cancer driver, and 60% of all the top 10 ranking nodes across all over 1,000 samples here are known cancer drivers. In uveal melanoma, we almost always identify a known cancer driver as the top node, less in the top 10. So here it drops a little bit more but mainly because there are fewer nodes which are fewer genes which are implicated as drivers in uveal melanoma. And we also have leukemia, so there the precision is not so high. But overall, this model can reproduce almost all the known cancer drivers across 32 of the 33 cancers. It does not seem to work very well for thymoma, but this is possibly because thymomas are not real cancers. But so thymoma is a little bit separate, but we can reproduce almost all the known cancer drivers using only these attention ranks. And we identify a number of novel candidates, which are either drivers or prognostic markers. They occur in a smaller number of individuals. So even if we have cohorts as large as 1,000 in breast cancer, it's built on Cancer Genome Atlas data. They are not significant across these cohorts, but this attention still ranks them highly in multiple individuals. And this is something that we can identify. So our attention mechanism seems to believe that this is the most important prognostic marker or driver for this individual. And this is something that we're currently investigating for some of these drivers. We can do other things with this as well. So you will have seen this in other cases. We can reproduce this relatively easy as well. So on the left-hand side, you see a TESDI plot. Each colored dot is essentially one sample. We can look at these samples to observe relations, similarity between patients. This similarity gives us a similarity in the subtype of the tumors. It gives us similarity in treatment response. It gives us similarity in survival time. On the right-hand side, it's simply a classification of cancers based on their molecular mechanisms, also produced based on the embeddings that our model has used. And finally, only 2 slides left. I know I'm a little bit late. So in the end, I like really this statement by Feigenbaum, and it's from one of the very first ICHCAI conferences. In Feigenbaum's paper, he stated that the domain-specific knowledge plays a critical role in organizing and constraining search. The theme is that in the knowledge is the power. This was intended by Feigenbaum for traditional search problems, so for discrete search, and it gave rise, or his paper gave rise to this search of expert systems. But more recently, this is again becoming more and more important, this time not for discrete search, but rather for continuous search and optimization problems in machine learning. And bioontologies are where we can find the background knowledge that we need in biomedicine. And with bioontologies, I explicitly include knowledge graphs, or most of the knowledge graphs, because I treat them as description logic A boxes. And the constrained optimization using large amount of relevant background knowledge allows us to go in our analysis to smaller and smaller cohorts, because the background knowledge constrains our search all the way up to single individuals, and therefore it allows us to move closer to precision health. It's not something that is without controversy, so this is all research, and these are all very active research areas. So I have a number of simplifications here, made a number of simplifications. So we use universal graph-based algorithms, but they are not always optimal for ontologies, because again, they don't rely on the a priori knowledge of the semantics or the model theory. And essentially, we need a more dedicated effort to study these relations between formal semantics and optimization, specifically in biomedicine. So in my group, we work a bit on homotopy type theory as a way to represent this, but there are many other ways. And the reason it needs to be specifically targeted to biomedicine is that biomedical ontologies are not structured like most of the ontologies or knowledge graphs that we find in general computer science applications. So not only are they mostly or very often inconsistent, but they have a number of other peculiarities. Okay, I really would like to thank my group at KAUST, so my students and my staff members, my collaborators, Paul and George, and my funders, and thank you very much.
Thank you, Robert. I think we have time for one quick question. There is one from the audience. Mega?
Hi, Robert. Thanks for the great talk. My question is regarding including this background knowledge. So in our own work, what we face when we try to incorporate this background knowledge in the form of protein interaction networks, for example, what we face is there are different kinds of data available, right? So one is from wet lab experiments, and there is a lot of data in gene ontology, for example, which is computational based, right? So which are from other computational models. And then also from this experimental data, we have the groups which deposited this data have different experimental settings, and they report certain confidence measures which are not comparable. Did you also face such problems, and what are your suggestions to overcome this? How to obtain a reliable set of data, or would you suggest, am I audible?
Yes, you're audible.
Yeah. Would you suggest to use everything that's available and it would have some kind of regularization effect? So do you have any suggestions in this regard?
Robert, you may unshare perhaps then.
Yeah. Robert, can you unshare your screen? Okay, didn't think he would. Ah, you're still. Yeah.
I'm sorry, my Teams died.
Okay. So shall I ask my question? I'll just quickly ask my question again. So my question.
In interest of time, I think we move on perhaps to the next speaker. There are a couple other hands up, and maybe they can contact you via email or Skype or something or other afterwards. Okay. Thank